In this video, I will explain different genetic phenomena and then provide an example for each one of them. The first condition is variable expression, which explains that the severity of the symptoms can vary in different individuals with the same genotype. For instance, mutation in NF1 gene can cause neurofibromatosis. So all patients with NF1 gene have neurofibromatosis, but the severity of symptoms between these individuals is variable, and this phenomenon is referred to as variable expression. The next condition is incomplete penetrance, which means that some individuals who have the disease genotype do not develop the disease at all. An example is BRCA1 mutation, where not all the patients with BRCA1 mutation will develop breast cancer. So some do develop, some don't. So that is referred to as incomplete penetrance, versus variable expression, where patients do develop the disease, but the severity of the disease varies from one individual to another. The next condition is codominance, where both alleles contribute to the phenotype. And a good example is blood type A and blood type B. So if two individuals mate with each other, one is with blood type A and the other with blood type B, their progeny will have blood type AB. So this is referred to as codominance because both alleles now contribute to one phenotype. The next condition is pleiotropy, where single gene mutations can develop multiple phenotypes. For instance, mutations in RET gene can cause multiple endocrine neoplasia, which can cause medullary thyroid carcinoma, as well as pheochromocytoma. So mutation in a single gene results in the development of multiple phenotypes. The next condition is polygeny, where multiple genes affect a single phenotype. So for instance, skin color is being regulated by multiple genes, not single one. So again, polygeny is a condition where multiple genes contribute to one phenotype, versus pleiotropy, where single gene contributes to multiple phenotypes. The next condition is anticipation, where in successive generations, patients will develop a more severe and early onset of disease. A good example is Huntington's disease. And males who have Huntington's disease, their progenies will develop expansion of the CAG repeats. And thus any individual, any progeny that have Huntington's disease will have earlier onset of the Huntington disease. And this phenomenon is referred to as anticipation. The next condition is imprinting, which explains that the symptoms depend on where the mutation is located, whether it's on the paternal or the maternal gene. And I would like to explain this phenomenon through these images. So as you can see, there are two genes that I've listed here, the Angelman gene and the Prader-Willi. Now what happens is that through methylation, these genes can become deactivated. So as you can see, Angelman gene gets deactivated in paternal chromosome, while Prader-Willi gene is being inactivated in the maternal chromosome. So by methylation, these two genes are no longer expressed. Now what happens is that if there is deletion of the maternal chromosome, or if there is paternal disomy, as a consequence of which there is no A gene effective in these patients, therefore they develop Angelman syndrome. Versus if there is a paternal deletion or maternal disomy, where the P gene will be absent, these patients will present with Prader-Willi syndrome. So again, the phenotypes of these patients depend on the chromosome that is being deleted. So paternal versus maternal can yield different symptoms. The next condition is dominant negative mutation, where the mutation of one gene interferes with the function of the other wild-type gene. So let's say for argument's sake we have two genes here, gene A and gene A. And let's say for argument's sake that the top gene undergoes a dominant negative mutation. Now this new protein will interfere with the function of the other one. And this phenomenon is referred to as dominant negative mutation, means that mutation of one gene interferes with the function of the other normal gene. Heteroplasmy is a condition where both normal and mutant mitochondrial DNA are present in an individual. So let me show you an example. These are mitochondria that are in cells, and the green ones are normal versus the red one, which is an abnormal. So these ones being transferred to new progenies, there would be a mix of normal and abnormal one. And this phenomenon is referred to as heteroplasmy. And the diseases that are associated with heteroplasmy are mitochondrial diseases, which include Leber hereditary optic neuropathy, also known as LHON, MELAS, which is referred to as mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes, and finally myoclonic epilepsy with red ragged muscle fibers. So just keep in mind that these three disorders are being affected by the heteroplasmy phenomenon. The next condition is mosaicism, where post-fertilization mutations make the cells in the body to have different genetic makeup. So this is due to post-fertilization mutations. And thus, in these patients with mosaicism, the genetic makeup of the nuclear DNA will vary between different cells, versus heteroplasmy, where the mitochondrial DNA is going to be affected. So let me give you an example of mosaicism. So this is an example of mosaicism where there was fertilization, the zygote was formed, and then later on there was a new genetic mutation after the fertilization, and thus some of these cells will continue to harbor the mutation, and thus parts of the body will develop the disease gene. Now a good example of mosaicism is in patients of Down syndrome, where they have triple copies of chromosome 21. So as you can see, there are normal chromosomes here that are normal 46, versus there are abnormal ones that are 47 that have triple 21 chromosome, making the whole genetic makeup 47 chromosome. So I would like to remind you that Down syndrome can happen through three different mechanisms. So the first one is non-disjunction of homologous chromosomes, which is commonly associated with increased maternal age. And this first condition accounts for 95% of the cases of the Down syndrome. The next condition is Robertsonian translocation, which is a rare form of translocation that affects acrocentric chromosomes, like chromosome 21. So this is chromosome 21 that have a small short arm and a long arm. So if one of these long arms get detached and goes attached to a different chromosome, and then later on during the cell division that chromosome is transmitted to the new individual, these new individuals will develop Down syndrome because they will have triple copies of chromosome 21. And this phenomenon is referred to as Robertsonian translocation, and it accounts for 4% of the cases of Down syndrome. And the next condition is mosaicism, where due to a post-fertilization error, these individuals can develop a different genetic makeup, and this accounts for 1% of the cases of Down syndrome. And so here we go again, mosaicism, post-fertilization error, these individuals will develop new cells that carry 47 chromosomes due to the extra copy of chromosome 21, and thus parts of the body will have trisomy 21. And that concludes our discussion.